still nasa first macromolecule pa tayo, but somehow almost end na po tayo. After we tackle these types of proteins, we will discuss enzymes, and that would be the end of proteins. And we'll proceed directly to nucleic acids. Okay? So for this afternoon, start tayo sa complex or the biologically active proteins. So yung main focus of our discussion is to be able to discuss two of the most dominant or most commonly observed proteins, no, in the body, and these are the fibrous, okay, and the globular proteins. Okay. Now, when we say fibrous proteins, these are important to provide tensile strength. Okay, I repeat, it is very important in the body. This type of protein kasi provides tensile strength. Okay, tensile strength. Ayan. And also, it is very important for flexibility. Ayan. So fibrous proteins, these are important to provide tensile strength and also flexibility. Whereas globular proteins in general, these are essential for metabolism, okay? Also very important for transport purposes. Okay, this is also important for transport purposes, and lastly for storage. Okay. Now, under fibrous proteins, we have the following. Although you have encountered this already, I have provided this, diba, sa atong preliminary discussion on proteins, the structure ng proteins, which is under ni sila sa tertiary structure. Fibrous proteins, we have collagen, elastin, and keratin. Whereas for globular, we have examples like myoglobin and hemoglobin. Okay, so let us begin with fibrous protein. And the first example under fibrous protein are the collagens, okay, or the collagen. Now, the collagen, this is an example of a structural protein. Okay, in fact, it is considered as the most abundant, the most dominant, okay, protein in the body. I repeat, this is the most abundant protein in the body, collagen. So ito yung pinakamaraming protein sa katawan, okay, which can be found on different connective tissues, okay, just like tendons, cartilage, okay, or in the cornea of the eye, or the matrices of the bones. Okay? With that, collagen is truly a fibrous protein because it provides tensile flexibility, and also it provides strength, no? It provides tensile strength, rather, and flexibility. Okay, di ba yung mga connective tissues na yan, tendons, cartilages ng bones, that provides structure, no, sa atuang cell framework. Okay, so it provides tensile strength. Yan yung collagen as a fibrous protein. Okay, now as a protein, it is also important to know what is the structure, no, the structure of collagen. Now, with regards to the structure, okay, structure ng collagen, it actually has a triple helix, okay, na structure. Okay, I repeat, it has a triple helical structure. Okay, that's why on the figure, diba? On the figure, you can see there are 3 chains that are intertwined. Okay? Ayan, di ba makikita naman may 3 chains that are coiled with one another. The one in blue, pink, tsaka yung nasa likod, yung kulay green. So, tatlong helices na nag-ikutan. Okay, so triple helical yung structure ng collagen. Now, ano tawag natin sa each pieces of that chains na naka-helical, okay, or naka-helix dyan sa figure or dyan sa structure ng collagen? Take note, ang tawag natin dyan per chain per chain dyan, ang tawag natin is alpha chain. Okay? I repeat, ang tawag dyan is alpha chain. Okay? Now, ano yung mga characteristics of an alpha chain? Ano yung mga characteristics or features describing ng alpha chain? Number one, take note, ang alpha chain, yes, isa siyang super helical structure. Okay? I repeat, isa siyang super helical, okay, structure. Okay? Now, when we say super helical, meaning to say, yung turning, yung turns, yung pagbuo ng mga liko, okay, yung pagbuo ng mga liko, mas madalas. Kaya nga, super helix, super helical. And that is, gaano kadalas? That is every 3.3 residues per turn. Okay, 3.3 residues per turn. Nabubuo yung mga turning, nabubuo yung pagliko. So that's why, again, super helical. Okay, yung isang feature ng alpha chain. Each of this alpha chain. Okay? Please take note, magkaiba yung alpha chain sa diniscuss natin na alpha helix Magkaiba yung alpha chain sa diniscuss natin na alpha helix. Recall that, may I ask you first, what level of structure yung alpha helix, if you can still remember? Ano nga ulit yung very good? Secondary structure. So magkaiba siya with the alpha chain that we are talking about for the structure of collagen. Okay, recall nga sa alpha helix, okay, every how many residues nabubuo yung turns sa alpha helix? Every how many residues? Very good. Every 3.6 residues, yan ang regular number ng residues para mabuo yung turn sa alpha helix. So comparing to alpha chain, less helical yung alpha helix. Okay, lesser helical yung alpha helix. Okay, now, when we check over the primary structure ng isang alpha chain. When we say alpha, I mean primary structure. Ano agad yung key term natin kapag primary structure yung pinag-uusapan? Ang makikita lang dapat ay, very good, sequence ng amino acid. Now, when we check over the primary structure of an alpha chain, meron siyang template. Okay? Pag sinabing template, yan yung standard format. Di ba? Pag sinabing template, yan yung standard format. Okay? So, si alpha chain ng collagen structure, meron siyang template na sequence. Okay? So, kumbaga, yan lang yung format sequence niya, paulit-ulit. Okay? So, sa primary structure ng alpha chain, we have gly, okay, X and Y. Yan lang yung paulit-ulit na mga residues na makikita sa alpha chain. Okay? Ng collagen structure. Gly there pertains to glycine. Okay? That pertains to glycine. Then, yung, before that, yung glycine, di ba, as we remember, it is the simplest amino acid. And that is considered simplest amino acid because of its side chain. Ano lang ulit yung nasa side chain ng glycine? Anong atom yun? Very good. Hydrogen. Now, because of that side chain hydrogen, simple acid yung glycine. And that gives it flexibility. Okay, that's the reason bakit flexible yung glycine in general. Okay? Diba sa preliminary discussion, I have mentioned na glycine, yung character niya, flexible yung glycine. Okay? Next, what does X here stands for? Yung X naman, this represents, it represents proline. Okay? Yung X dyan, proline. Now, what is the importance of proline sa alpha chain structure? Take note, it is the proline that provides kinks. Okay, it provides kinks in the structure of the alpha chain. So, yan yung importance ni proline. Okay, it provides kinks. And finally, we have Y. Yung Y dyan, that could either be, yung nasa parenthesis, that could either be HYP, which stands for hydroxyproline. Okay, that's hydroxyproline. Or that could either be HYL. HYL stands for hydroxylysine. It's not leucine, it's lysine. Okay? Hydroxylysine or hydroxyproline. Yan lang yung letter Y. So again, that is the template, no, format sa primary structure ng isang alpha chain. Okay? Sa collagen structure. Okay, question. Based on your knowledge on amino acids, are these HYP, HYL standard amino acid or derived amino acids? Very good. These are derived amino acid. Meaning to say, galing sila sa standard amino acid. Now, ano ngayon yung importance ng hydroxyproline tsaka hydroxylysine? Okay, take note, sila naman yung nagpo-provide ng tensile strength. Okay, they provide tensile strength. Okay, so ang point dito is, kahit gaano pa kahaba yung collagen structure, kung titingnan natin yung primary structure nito, ang amino acid sequence paulit-ulit lang, and that is gly, X, and Y lang yung makikita. Yan lang yung sequence amino acid na nakikita natin. Okay? And again, yung Y dyan, that could either be hydroxyproline or hydroxylysine. Now, based on your knowledge or your preliminary knowledge na discuss ko sa inyo, which of the two kaya ang much more common na makikita sa isang collagen or very important for collagen synthesis? Very good. It's the hydroxyproline. So take note of that, ha. Between the two, ang pinaka-common dyan na letter Y is hydroxyproline. Okay? Now, another figure, ayan, that discusses structure of amino acid sequence of an alpha chain, okay, ng collagen structure. So, eto. So, another lang niya siya, the same lang gihapon nga concept that I have discussed sa previous slide. Paulit-ulit lang. Yung amino acid sequence gly, XY, gly, XY. So, gly ulit, this is for flexibility. Okay? For flexibility. Yung X, yung proline, that is very important for kinks, formation of kinks. And then yung Y dyan, that could be HYP, HYL for tensile strength. Okay? Tensile strength. It provides tensile strength sa structure ng collagen. Okay? Then for its secondary structure, recall, sabi kanina, 'di ba, yung collagen, it has a triple helical, no, na structure, and alpha chain. Okay, yung first feature na diniscuss ko, super helical, no, super helical yung structure niya. So that's for the secondary structure, super helical. Okay? Ayan. So kumbaga, kapag super helical, madalas, no, madalas yung mas madalas nabubuo yung turning, yung turns. And that is again every 3.3 residues per turn. That's for the alpha chain. Okay? Then you have here the triple helix. Okay, triple helix structure. So yung sinabi ko kanina, tatlong alpha chain. Okay, yung per chain dyan, alpha chain yan. So, tatlong alpha chain, okay, na nagko-coil, naga-intertwined with one another. Okay, yan lang yung triple helix. Okay. Now, for its nutritional value, we actually have nutrients, no, in our diet, in the food that we eat na rich in collagen. Example, yung gelatin. Familiar kayo sa gelatin na dessert? Okay, that is rich in collagen. Yung capsules, yung mga gamot na enclosed in capsule form, okay, mapa-soft gel or yung dried capsule, that is also rich in gelatin. That is actually made up of gelatin. So its component, therefore, kung titingnan natin yung primary structure nila, yung makikita dyan would be gly, X, and Y. So collagen rich. Okay? Yung gelatin. Okay? Now, also, meron tayong mga vitamins, di ba? Maraming marketed, not really vitamins, yung supplements pala. Okay? Maraming supplements, no, available in the market na kiniklaim nila, sabi, rich in collagen daw yan. Okay, so bili ka nito, rich in collagen ni nakakaganda daw ng balat. 'Di ba? Nga na ilahang pag-advertise kapag rich in collagen, makagwapa ni siya, muinat gyud imuhang nawong. Ay grabe pod muinat. Muinat ang panit, dili ang nawong, okay? So if you happen, no, to be gihatagan ka ana, gi-advertise na sa imong friend, paliti ko ani friend, akong advice sa inyo ha, ayaw magtuo. Okay? Dili na siya tinuod actually. Okay? Scientifically speaking, hindi po yan totoo nga gina-claim nila nga gapagwapa ang collagen, gapagwapa og balat. Og balat ha, dili gyud ang itsura ma-change ni mo itsura. Gapakuan siya ba? Inat siya og balat para mo gwapa, mo bata balik ang imuhang skin. Okay, so ayaw magtuuan nga claim, hindi po yan totoo. Okay, nga sabi nakakapaganda ng balat. Okay, the reason there is, remember, di ba, ang collagen, sabi ko, on its primary structure, yung makikita nga amino acid present are only gly, XY. Now, try to imagine yung kiniklaim nilang supplements na rich in collagen. Okay? Siyempre, supplement yan, so that would be available in tablet or in capsule form. So, unsaon man na pag-inom, siyempre, via mouth, di ba? So, di man na pwede inject, supplement, ang katablet man siya. So, it has to be administered. It has to be taken by mouth, kung ingana. Okay? Siyempre, kapag taken by mouth, listen, kung taken by mouth yung isang supplement, isang substance, siyempre dadaan yan sa stomach. And sa stomach po natin, sobrang dami, maraming hydrolytic enzymes. Okay? Sobrang daming hydrolytic enzyme. And pag sinabing hydrolytic enzyme, anong reaction yung gagawin ng hydrolytic enzyme? It causes hydrolysis, no, nga reaction. And please take note, kapag may hydrolysis reaction, ang tendency, etong bonding ng mga amino acid, XY, ang tendency, those bonds will break. Okay? It will be destroyed with hydrolysis reaction. Okay? Yan yung isa sa mga way, di ba? Para from peptide chain, para mabuwag-buwag natin yung mga individual amino acid in that peptide chain, hydrolysis reaction yung i-apply. So kapag na-exposed yung sinasabi nilang rich in collagen na supplement sa stomach, maraming hydrolytic enzyme, makikleaved lang din, no? Makikleave lang din yung bonding ng residues, okay? Present in the primary structure ng collagen. So, ayaw magtuo nga, yes, rich in collagen nga yan, pero again, dili na siya full claim nga nakakaganda po siya ng balat. Okay? Kanang mga endorser nila, gwapa na gid na sila daan. Dili na tungod sa ilang gipang inom. Okay? Nga rich in collagen. Okay? Next. So, yun ang scientific, no? Scientific na explanation. So, it's up to you, no? If you buy that, okay? Wala man po na siya problema, no? Kung mutuota. But at least you know already the scientific reason na yung kiniklaim nila hindi yan full, no? Fully proven, okay? Next, with regards to hydroxyproline, okay? Ayan, hydroxyproline na po tayo. Diba? I have mentioned, very important din to sa primary structure ng alpha chain of collagen. Okay? Now, with regards to hydroxyproline, which is a derived amino acid, okay, as mentioned, it is again very important for tensile strength. Okay? Diba? This is the one very important to provide tensile strength. Now, discuss natin paano po nagagawa, okay, or paano nasisynthesize yung hydroxyproline. Okay, so apparently, again, galing siya sa proline nga. Diba? Galing siya sa proline na standard amino acid. So this derived, no, amino acid that is being produced from hydroxylation reaction of proline residue. Okay, hydroxylation ha, not hydrolysis, magkaiba yan. Hydroxyl OH, okay, addition of OH, hydroxylation of the proline residue. So that will form hydroxyproline. Okay, and take note, the responsible enzyme for the hydroxylation of proline is the one highlighted. It's the prolyl hydroxylase. I repeat, kailangan ng enzyme to catalyze hydroxylation reaction of proline, converting it to hydroxyproline, and that is via the enzyme prolyl hydroxylase. So ang point lang po dito is from proline, mula proline, okay, via this enzyme prolyl hydroxylase, that causes formation of hydroxyproline, HYP. Okay? However, take note, etong prolyl nga sinasabi ko, prolyl hydroxylase na enzyme, okay, this will only be activated, okay, or magiging useful lang siya dyan sa catalytic reaction na yan in the presence of cofactors. Again, in the presence of cofactors. Yung cofactors kasi, by definition, kailangan yan ng mga certain enzyme para ma-activate sila. Diba? Enzyme are very important for catalytic reaction. Now, we have certain enzymes na hindi sila nakaka-catalyze ng reaction without cofactors. So, cofactors, kailangan nila, needed yan para ma-activate, magkaroon ng usefulness yung enzyme. Okay, so halimbawa ng cofactors na kailangan ni prolyl hydroxylase dito are the following. Okay?
We have oxygen, the ferrous ion, and vitamin C. Vitamin C, that is also known as ascorbic acid, or this is also known as sevitamic, no? Sevitamic acid. Ayan. So those are just synonymous na mga terms with vitamin C. Sevitamic acid, ascorbic acid. Okay, so I repeat, these are the cofactors. Now, with the presence of these cofactors, yung prolyl hydroxylase can now catalyze proline into hydroxyproline. And via what reaction? Hydroxylation. Okay, so that is now the reason, because of the formation of hydroxyproline, we have that already provided strength to the collagen structure. Okay, this is the reason that somehow strong, no, yung characteristic ng collagen. Okay, ayan. Next, I'd like to emphasize also with regards to vitamin C. No? Okay, with regards to vitamin C, of course, we all know na this is a very common vitamins, di ba, na iniinom ng tao. Okay, and sobrang daming drugs available sa market containing vitamin C. Now, sa sobra kadami ng mga brands ng vitamin C available sa market, syempre, kanya-kanya ang mga kapulo, di ba? Na sila kanya-kanya na kapulo how to market their brands. And mostly, ang gina-advertise din kapag ascorbic acid, it boosts immunity, that is very true. Okay, very important siya for immunity. Okay, but we actually have a certain brand ng vitamin C na sobrang galing yung pagka-advertise. Okay, galing ng pagka-advertise nila. Dili lang nila i-emphasize na it can boost immunity. Okay, you can search on the internet, yung commercial ni Miss Bea Alonzo. Familiar mo itong iyang i-advertise ng vitamin C, the brand Potency. Familiar man siguro ka sa brand na Potency. Okay. Tsada, no, ilahang sobrang galing ng pagka-advertise nila kasi aside sa binigyan nila ng halaga yung immunity, boost immunity, that is very common, ang iya pag yung giingon ni Ms. Bea, nakakaganda ng balat si vitamin C. And that is very true. Okay, very true po nga si vitamin C, okay, nakakaganda siya ng balat. Okay, vitamin C, nakakaganda yan ng balat. Okay, so I repeat, vitamin C is a cofactor, di ba? Cofactor siya para mas dumami yung hydroxyproline, para mas mabuo, no? Mas synthesize natin yung hydroxyproline. And again, it is the hydroxyproline that provides strength sa structure ng collagen. Okay? So that is why collagen-rich skin, kapag collagen-rich na skin, babanat mo inat yun ang imuhang skin, ang imuhang okay, I'm sorry for that. Huh? Sige, I repeat, kapag ang skin collagen-rich, okay, feeling young looking good kaayo ang skin mo, inat na na. Okay? That's the reason. Kasi nga, may tensile strength, no? Yung structure ng collagen. And yung claims nga na nagsasabing nakakaganda yung vitamin C, yan ay totoong claim. Okay? And yan yung scientific explanation dyan. Okay? Kasi pag marami yung vitamin C with increased vitamin C intake, di ba important ito as cofactor, okay, ng enzyme prolyl hydroxylase. At samot na mudaghan ang mabubuong hydroxyproline, and that is very important for collagen na synthesis. Okay? So, kamo magainom og vitamin C, okay, very good yan na practice. Okay, very important din yan for our skin. Nakaka-increase niya yung collagen production. And collagen is very important for skin. Next, let's go back to hydroxyproline. Okay, going back to hydroxyproline, again, it stabilizes the triple helical structure ng collagen. Okay? Ayan. Now, for this table, kabisaduhin nyo lang to. Okay? You simply read this. Okay? Ang point lang diani, this table, it only states the different types of collagen. Different types, type 1, type 2, type 3, type 4, etc. Okay? And saan sila makikita sa body. Okay? So as being shown in the table, di ba, sobrang dami ng parts ng body kung saan makikita yung collagen, making it the most abundant protein in the body. Okay? So kabisaduhin niyo na lang yan. Okay? Kung type 1, saan siya nakikita, etc. Okay? Next. At this juncture, let us discuss the biosynthesis of collagen. I repeat, let's have a more detailed discussion on how collagen is being synthesized. Okay, so in here we will understand how collagen forms the triple helical structure. Okay, paano nabubuo yung triple helix niya na structure or triple helical structure. Okay? So we can actually divide the steps into 7. Ito yung main na steps. Although pagdating sa individual na discussion, na siya 9 kasi may mga sub steps na i-discuss natin. Don't worry, i-discuss natin po individually para mas maintindihan paano nabubuo yung triple helix structure ng collagen. Okay, so we'll start with with step 1, yung pro-alpha chain formation up until cross-link formation. Okay? So, i-discuss, no worries, i-discuss natin yan individually. So, this figure naman, itong figure na to, it only summarize lang ito, it only summarizes the different steps involved in collagen formation. Okay, so starting with the gene that encodes the collagen until such time na may cross-linking. Okay? Sige, so iisa-isahin natin yung each steps involved, no, in this process. Okay. So let's have the first step. Okay, number 1, it involves the production of the so-called pre-pro alpha chain, okay, on the ribosomes. I repeat, first step is the production of the pre-pro-alpha on the ribosome. So how does it happen? Okay, yung synthesis ng said chain, okay, sa ribosome. Suppose this is the nuclear membrane. As labeled, you have there the nuclear membrane. Okay, so Syempre, ang nuclear membrane, saan man niya nakikita, di ba, it encloses the nucleus. Di ba? So, here's the nucleus. Then, yan yung covered niya na nuclear membrane. Tandaan, inside the nucleus, mRNA, or remember, the ribosomal biosynthesis happens in the nucleus. Okay, in the nucleus, nangyayari yung ribosomal biosynthesis. So, halimbawa, doon na-synthesize yung mRNA. Now, this mRNA, siya ang magiging transcript. Okay? I repeat, siya yung magsiserve as transcript. Script. Okay? Or siya yung magiging basehan or guide para gumawa ng isang protein. So in this case, what protein that we are tackling, gagawa tayo ng collagen. Okay? So yung magiging basehan ulit ha, mRNA. Okay? In order to synthesize the protein, specifically yung protein dito na dinadiscuss is collagen. So for instance, we already have mRNA. So meron ng mRNA. Okay? Na na-produce, of course, sa nucleus. Now, this mRNA will go out. Okay? It will go out. Okay? It will exit. Okay? The nucleus. Okay? So from the nucleus, i-exit siya via, di ba may mga pores sa nucleus? Ang tawag doon, nuclear pores. Okay, so i-exit siya sa nucleus via nuclear pores. Hanggang makarating siya dito sa ribosomes. Okay, so you have mRNA na na-produce sa nucleus. It will exit the nucleus via nuclear pore. Papupunta siya sa ribosome. Now, in the ribosome, what happens here is translation happens. Okay? Translation will occur in the ribosome. When we say translation, yung mRNA transcript, yan ay magiging kopyahan, copy, para makagawa tayo ng isang polypeptide, isang protein, diba? Polypeptide. Kaya ang tawag natin sa polypeptide na mata-translate dito, okay, is what we call the pre-pro-alpha chain. Okay? I repeat, okay? Sa ribosome, dito mangyayari yung translation. Okay, translation will occur kung saan yung mRNA na to magiging transcript, kopyahan siya para makagawa tayo ng polypeptide. And ang tawag sa initial polypeptide na mata-translate, pre-pro-alpha chain. Okay, so ang pre-pro-alpha chain dyan, yan yung kulay red to blue. Red to blue nga line direct. Okay, so parang ito yun. Yan na siya. Diba na kay Ana, red, then na kay somehow yung kulay blue. Muna siya ang whole. Okay, yung whole line dyan consisting of red, blue as shown in the figure. Yan yung pre-pro-alpha chain. Okay, next let me highlight this signal peptide being labeled. Take note. Generally, or essentially speaking, kapag ang isang polypeptide chain, by the way, yung kulay blue dyan, yan yung being labeled ha, as the signal peptide. Okay, but again, yung whole dyan, yung whole structure, yan ay isang polypeptide. Ang pangalan natin dyan is the pre-pro-alpha chain. Now, I will just highlight what is that signal peptide. Okay, take note. Kapag ang isang polypeptide, okay, merong signal peptide, ang ultimate destiny anak ng polypeptide or ultimate fate niya ay ilalabas siya sa cell. Okay, it will exit the cell. Okay, I repeat, kapag ang isang polypeptide merong signal peptide, yung ultimate fate, okay, is that it exit the cell. Okay? Di ba naalis siya sa cell? Kaya na may ribosome. Asa man ang ribosome? Di ba na siya sa usa ka cell? So, ang point dito, kung ang mabubuong polypeptide mo sa ribosome kapag walang signal peptide, usually naiiwan lang sila sa loob ng cytoplasm of the cell. Now, in this case, di ba, may signal peptide man, sa pre-pro alpha chain, okay? So, lalabas siya sa cell. Okay, kumbaga sa signal peptide, siya yung magsisilbing driver, no? Driver ng nabuong polypeptide para ito ay mailabas sa cell. Okay, next. Okay, next step. Next na tayo, hayan lang yung concept sa number 1, step 1. We have to synthesize the pre-pro alpha chain and saan siya nabuo? Sa ribosome. Okay, next. As mentioned earlier, sabi ko diba, ang signal peptide, siya yung magsisilbing driver, okay, ng peptide chain para mailabas yun sa cell. So, ano yung susunod na mangyayari kapag nabuo na yung pre-pro alpha chain? Okay? Now, the next step, okay, is that itong signal peptide, okay, which is highlighted with kulay blue, okay, diba? It is the one that will direct the polypeptide chain first to the RER. So, diba, driver siya, dadalhin niya yung buong pre-pro alpha chain mula sa ribosome papuntang RAF-ER. Okay? And ang purpose dyan, bakit niya i-direct papuntang RER, RAF, endoplasmic reticulum, ang buong polypeptide chain para mabuo yung pro-alpha chain. Okay? Now, what happens here is, from initial na pre-pro, diba? Initially pre-pro yan. Pre-pro alpha chain. Saan nabuo? Ribosome. Okay, so from the initial pre-pro alpha chain na nabuo sa ribosome, kapag pumasok na yan sa RER, nakiklive. Okay, nakiklive yung signal peptide. Matatanggal yung kulay blue dyan. Matatanggal yung signal peptide. So when that signal peptide portion is being cleaved, ang tawag na sa natira, ang tawag natin sa natirang chain ay pro-alpha chain. Okay? Pro-alpha chain na lang yan kapag natanggal yung signal peptide portion. Okay? So take note, kapag pre-pro, may signal peptide pa yan. Kapag pro-alpha chain, natanggal na, no? Natanggal na yung, na-cleave na yung signal peptide. Okay. Moving on. Nasa RAF-ER na po tayo, ha? Nasa RAF-ER. Okay, or the RAF endoplasmic reticulum. And I repeat, pro-alpha chain, diba? Pro-alpha chain yung andito. Now, what happens here? Take note, sa isang pro-alpha chain, okay, sa isang buong pro-alpha chain, may residue dyan na ini-improve. Okay? May residue dyan na ini-improve. Okay, go to the next slide. Okay? I repeat, meron ka ng pro-alpha, ayan, pro-alpha chain na nabuo ulit sa RER. Ngayon, merong isang residue sa buong pro-alpha chain na kailangan natin i-improve, kailangan natin i-convert into a stronger version. And that involves hydroxylation of lysine, okay, and also hydroxylation of proline. Okay, so for lysine, halimbawa, for lysine, via the enzyme lysine hydroxylase, that results to hydroxylysine. Whereas, for proline, okay, para ma-improve natin si proline into a stronger version, via the enzyme proline hydroxylase, we will form hydroxyproline. Question, may cofactors needed ba sa enzyme na prolyl hydroxylase or proline hydroxylase para ma-activate itong enzyme na to? May kailangan ba tayong cofactors? Very good. Yes, we have cofactors. And ano nga ulit yun? We have oxygen, ferrous, at saka vitamin C. Okay, so please take note ha, nangyayari itong conversion na to, okay, sa RER pa rin, sa RER. Diba? So imagine niyo lang sa loob ng RER, may improvement sa mismong one of the residue ng pro-alpha chain. And yun na nga, yun yung lysine, converting that to hydroxylysine, proline naman into hydroxyproline. Now, next step, step 4. Recall muna before ka mag glycosylation. Okay, synthesis, di ba, yung unang step. Okay? Step 1, synthesis of pre-pro-alpha chain. Second step, naging pro-alpha chain na. And third step, naanatay, derived amino acid formation. Okay? So nabubuo yung HYP, nabubuo yung HYL. So yan yung first 3 steps. Now at this juncture, let's proceed to glycosylation. Step 4 na ha. Now, when we say glycosylation, there is actually addition of carbohydrates here. Okay? Sa mismong structure ng alpha chain, there is carbohydrate. CHO means carbohydrate. Okay? CHO, that's carbohydrate. Yan lang kasi yung component ng isang carbohydrate. Carbon, hydrogen, oxygen. So CHO, there is carbohydrate addition in the structure. Okay? So addition of particularly the lactose or either glucose to some hydroxylysine residue. So please take note, yung addition of carbohydrate usually happens sa hydroxylysine residues. Okay, so kapag hydroxylysine, okay, yung present na residue sa primary structure ng collagen. So yan yung nangyayaring addition or glycosylation. Okay, so again, it's either galactose or glucose na carbohydrate. Kaya may nakalagay dyan, di ba, na enzymes involved, galactosyl for galactose, glycosyl transferase naman na enzyme kapag glucose yung ina-add. Okay? So, ganun pa rin itong process na to, nangyayari ito sa RER, rough endoplasmic reticulum glycosylation. Okay, next. Proceed tayo sa step 5. Okay, so at step 5, there is already assembly of 3, pakidagdag na lang, that's pro, assembly of 3 pro-alpha chain. Di ba pro-alpha chain tayo yun, yung nabuo natin? Okay, so assembly of the 3 pro-alpha chain to form the pro-collagen. Okay, so as stated, this involves formation, involves formation of disulfide bonds. It involves formation of disulfide bonds between the C-terminal extension of the pro-alpha chains. Okay, so for step 5, there is an assembly, take note, there is an assembly of or alignment, okay, or pagtitipon-tipon ng pro-alpha chain. Okay? And please take note that happens in the Golgi apparatus already. The assembly of protein sa Golgi apparatus yan, di ba? So I repeat, itong assembly process na to nangyayari sa Golgi apparatus. So ibig sabihin, kapag assembly of 3 pro-alpha chains, so say for instance, you have here, ibig sabihin lang yan, meron kang tatlong pro-alpha chain. Okay? Okay. Yung tatlong pro-alpha chain, mag-a-assemble sila, magsasama-sama sila. Okay? Yan yung nangyayari sa step 5. And ang distinct dito, as being stated, there is presence of disulfide residue sa C-terminal portion. Okay, sa C-terminal portion, merong disulfide residue. Ano ba yung disulfide, diba? Disulfide, you have a sulfur bonded to another sulfur. Ayan. Yan yung, hindi lang siya klaro, but ito yun. Okay, you have SS, diba? Nakakita SS. So yan. Sa dulo, sa C-terminal, take note na sa C-terminal, you know already where is the C-terminal, that is on the rightmost end, di ba? So sa C-terminal daw po, merong presence of disulfide residue, okay? Na, take note, yan yung nagfo-form, okay, ng pro-collagen. Okay? I repeat, sa assembly of 3 pro-alpha chain, okay, ang distinct diary, okay, sa C-terminal, meron tayong makikitang disulfide residue. Sa C-terminal, merong disulfide residues. Okay? And then, ang ultimate goal dito, mafo-form yung pro-collagen. Diba? Kapag nag-assembly yung tatlo, mafo-form yung pro-collagen. Now, ano ba yung feature, hitsura ng isang pro-collagen? Pro-collagen already. Okay, ano ba yung hitsura? Okay, sa centermost ng, ako na lang mag-draw. Pwede man maklaro ang figure. Okay, sa centermost ng pro-collagen, halimbawa, there is itong tatlong pro-alpha chain, okay, magko-coil na yan sila sa centermost. So, halimbawa, ito yung center part. Okay, this is the first chain. Di ba, tatlong chain yan. 3 pro-alpha chain. Mag-assemble. Okay. Magko-coil yan kasi nga meron kang disulfide bond sa C-terminal. Okay. So, sa centermost, may coiling. And ito yung pangatlo. Hindi lang siya klaro. Basta ka-imagine niyo na lang sa centermost, mag-coil na sila. Okay? Or ingani nga coiling. Free na siya. Free ka chain nag-coil sa centermost. Okay? So, yan yung isa sa feature, no? Ng isang pro-collagen. Okay? We're talking about pro-collagen, ha? Pro-collagen. Sa center ulit, sa center portion, helical. Helical na po. And then, sa dulo naman, sa terminal end niya, take note, non-helical. Okay, kumbaga parang ganito. Wala pa siya helix. Ana lang. Then, sa dulo din, ana. Okay? So, I repeat, kapag pro-collagen, yung feature sa centermost, okay, sa center, helical. Okay, sa center, H for helical. Then sa lateral portion niya, non-helical. Okay, non-helical. Non-helical. Okay, so yan yung hitsura. Okay, if you try to imagine the pro-collagen na nabubuo upon assembly of this 3 pro-alpha chain. Okay, na merong disulfide bond sa C terminal. Okay? Ayan. So, kato akong i-discuss, maura po ni. Okay, ito lang yun. Okay, so as being stated, di ba? You have their 3 alpha chain. They are wound around one another. They wound, meaning nag-coil, nag-intertwine around with one another, okay, in the form of triple helix. But again, on the center portion pa lang ha, sa center portion pa lang yung nag-coil. Yan yung pro-collagen structure. And by the way, yung tawag natin sa non-helical sides, diba, may mga non-helical na portion. Ang tawag dyan is registration peptides. Okay, registration peptides. So, yan lang yung concept sa step 5. Okay, meron kang tatlong pro-alpha chain. On their C-terminal may disulfide bond. Yan yung reason mako-form yung pro-collagen. Now, ano ba yung itsura ng pro-collagen? Sa centermost, helical. Sa terminal ends niya or sa lateral portion, non-helical. Ang tawag ulit sa non-helical portion, registration peptides. So, done with step 5. Okay? Sige, recap ta sa gikan sa step 6. Okay, gikan sa step 6, step 5 kung dito, no? Gikan sa step 4. Okay, step 4 man eh. Or step 3 ba yun? Oh, step 3. Okay? Hydroxylation, yun yung step 3, di ba, na na-discuss. So recall, prior to step 5, prior to assembly, di ba, this is already step 5, assembly. Merong nangyayaring hydroxylation. And then, followed by glycosylation. Question, saang organelle nangyayari yung hydroxylation tsaka glycosylation? Anong organelle ng cell? Very good. It is in the RER, rough endoplasmic reticulum. And yung, it will be followed by assembly. The assembly of protein. Okay? It will happen naman sa Golgi apparatus. And ang inyuhang huna-hunaon ha, there is sa assembly, mabubuo yung pro-collagen. You take note ha, you take note what is the feature of pro-collagen. Sa center part, diba? Sa center part, helical, may helical sa centermost, and then on the lateral portions sa terminal ends, non-helical. O anong tawag doon sa non-helical portion? Anong tawag sa non-helical portion? Very good. Registration peptides. Very good. Registration peptides. So yan yung itsura ha. If you try to imagine the pro-collagen. Sige, proceed na ta sa step 6. Okay, step 6. It already involves exocytosis. Okay, so sulat nyo, this already involves exocytosis. Okay, for instance, by the way, when we say exocytosis, yung nangyayari dito, halimbawa, this is the Golgi apparatus. Kasi di ba, asa nabuo yung pro-collagen sa Golgi apparatus. Doon yung assembly, di ba? Assembly. So doon probably, doon natin makukuha yung pro-collagen. So, ngayon, sa Golgi apparatus, parang ganito man yung itsura ng Golgi apparatus. Ang mangyayari, ipapack. May packaging na mahitabo, di ba? Sa Golgi apparatus, there will be packing, okay, ng mismong protein, okay, na nabuo doon. There will be packing of the pro-collagen into vesicles. Okay, ipapack siya into vesicles para ready na siya mailabas through exocytosis process. Kapag sinabi kasing exocytosis process, that is the release, no? Release of contents from a membrane or from an organelle. So, yun yung ginagawa ngayon ni Golgi apparatus. Nirelease niya in a form of vesicles yung pro-collagen na pinak niya, yung pinackage niya. Okay? So, at the end of the step 6, at the end ng step 6, we can really tell now na, di ba, na-release na siya sa Golgi apparatus. So, we can tell now that the pro-collagen is now found on the extracellular space. Kasi nailabas siya ng Golgi apparatus. So, na siya, available na yan sa extracellular space or extracellular matrix. So, yan lang yung concept sa step 6. Exocytosis, secretion, excretion, release of pro-collagen by exocytosis para available na siya sa extracellular space. Yan lang yung concept sa step 6. Next. Okay, step 7 na tayo. Okay, so step 7, this already involves cleavage. Okay, cleavage of registration peptides. Okay, question. Ano nga ulit yung registration peptides? Helical, non-helical. Very good.
Non-helical.
Very good. Non-helical. Now, in the extracell, di ba, nasa extra na tayo. Nasa extracellular space na tayo. Okay? Now, sa extracellular space, yung nailabas na pro-collagen, okay, it will be catalyzed by an enzyme. Okay? It will be catalyzed by an enzyme which is called pro-collagen peptidases. Okay? Kasi kapag nasa cellular space na yan, extracellular space, marami ang mga available enzyme din. No? So, isa dyan that will catalyze the pro-collagen, okay, is the enzyme pro-collagen peptidases. Now, for you to understand this, just take note that kapag sinabing peptidases, that is another example of hydrolytic enzyme. So, anong reaction kapag hydrolytic enzyme? Anong reaction yan? Anong reaction kapag hydrolytic enzyme? Very good. Hydrolysis. It promotes hydrolysis. And take note, kapag hydrolysis na yung reaction, it really destroys peptide bonds. No? Kaya nga peptidases, it will catalyze, it will break peptide bonds. That's bonds. Peptide bonds. May space dyan. Okay, peptide bonds. So, with pro-collagen peptidase, it will cause cleavage. No? Sisirain niya ang peptide bond, okay, that links registration peptide to the helical portion. Di ba sabi ko kanina, itong nasa drawing, huwag yung i-gamitin yan as guide ng pro-collagen ha. Ang pro-collagen, di ba, is dapat sa lateral portion, sa terminal portion, dapat hindi pa helical. So, halimbawa, ayan, ito yung mga lateral portion. Yung gitna, di ba, helical. Yung nasa dulo, non-helical, non-helical. Okay, ang non-helical, yan yung registration peptides, diba, sabi natin. So, yung gagawin ni pro-collagen peptidases, okay, isisirain niya yung peptide bond na nagko-connect, nagli-link sa registration peptides to the helical portion or centermost portion ng pro-collagen. Kaya syempre, di ba, what connects amino acid residue peptide bond? So, sisirain niya yung peptide bond dyan. Okay, that's why, as you can see on the slide below, okay, with pro-collagen peptidase, ang natira is only the centermost, no? Yung natira is yung centermost, yung helical portion, okay, ng pro-collagen collagen. And take note that resulting molecule, after catalytic reaction ng pro-collagen peptidases is what we call tropocollagen. So please take note of the different terms that we use. Mula pre-pro-alpha chain, pro-alpha chain, pro-collagen, tropocollagen. Okay? I know you understand, no? Mastery lang siya. I-master lang ninyo kailan siya pro-collagen, kailan siya tropocollagen. Okay? So, yan lang yung concept naman sa step 7. Kapag na-cleave, of course, kapag na-cleave yung registration peptides, tatanggalin siya. No? Tatanggalin siya from the pro-collagen structure. Yung matitira, tropocollagen yung tawag. Okay, sige. Step 8 na po tayo. Ayan. Okay, step 8. This involves self-assembly. Okay, self-assembly or polymerization ng tropocollagen. Okay, yung tropocollagen molecules nagpo-polymerize. Okay, forming collagen fibrils. So in this case, there is overlapping lang po. As you can see, no? Nag-overlap yung mga tropocollagen. Okay, overlapping of the neighborhood by a length approximately 3/4 of a molecule. So as you can see here, there is overlapping ng tropocollagen, forming fibrils. Okay, kumbaga, nagne-networking na sila ba? The network, parang may interconnection, and that will further strengthen the collagen. Okay, the collagen in a fibril structure. Okay, and lastly, step 9, last na to. Okay, lastly, we have cross-link. Okay, cross-link formation. So in cross-link formation, to be able to facilitate cross-linking, okay, or what I mean here is para mas maging more stabilized or more stable ang collagen na mabubuo, cross-linking will happen. Okay, cross-linking will happen para mas stabilize, mas more stronger, okay, yung mismong collagen na mabubuo. And please take note that in cross-linking, we need the enzyme lysyl, highlight ko ha, lysyl oxidase. Okay? Ayan. Okay? Ayan, nakasulat naman siya. Okay, so fibrillar array of collagen molecule that will serve as the substrate ng lysyl oxidase. Okay? And please take note, the same with yung prolyl hydroxylase. Okay, the same with prolyl hydroxylase. Si lysyl oxidase dito, in order to be activated, okay, kailangan niya din ng cofactor. Okay, kailangan merong cofactor. Okay, and as you can see, the ion, okay, na kailangan niya as cofactor to be activated is the cupric ion or the Cu2+. Okay, the copper ion, copper 2+. Okay, so that is the concept for cross-link formation. Kumbaga, ito na yung last step. So, it will now form a collagen, a much more stable, a much more stabilized, no, na structure of collagen, and that is, kailangan natin yung lysyl oxidase para may cross-linking, okay, na ma-form. Okay, and please take note, kailangan niya ng cofactor. The cofactor is copper ion. Okay? Now for this figure, this actually summarizes what we have discussed earlier from step 7, okay, to step 8. Okay, step 7 to step 8, parang summary lang din ito. Okay, di ba recall, yung pro-collagen, di ba, mula sa pro-collagen, di ba, meron siyang helical portion sa centermost. Meron siyang helical portion sa center and non-helical, okay, non-helical sa lateral portion yan. Now, itong non-helical portion, they are being cleaved. Okay, they are being cleaved, okay, via the enzyme peptidases. Diba? We have the pro-collagen peptidases as mentioned sa earlier slide. So yung matitira ito, and yung tawag dyan is tropocollagen. Okay? And this tropocollagen, it will cross-link. Okay? Magne-network sila. Okay? Magsa-self-assembly sila yung mga tropocollagen, okay, into fibrils, no? Para maka-form ng more mature collagen fibers. Okay? So, summary lang yan for step 7 to 8. Ito, I don't know if maklaro siya. Ito, summary, hindi na maklaro, no? Sige, let's try daw. Wala siya, maklaro siya, slide. Halap-halap lang din siya. Halap-halap siya, pero somehow mabasa siya slide, no? Sige lang. Summary lang naman to. Summary lang ni siya sa buong processes or buong biosynthesis ng collagen. Diba? Recap from step 1. Okay, from step 1, merong mRNA. Diba? Sa step 1, merong mRNA. Saan mapupunta yung mRNA? Mapupunta siya sa ribosome. The tendency, ma-form yung pre-pro-alpha. That's pre-pro-alpha chain. Okay, pre-pro-alpha chain. Okay. And then from ribosome, sige lang, i-check. Kung ano siya ha, summary. From ribosome, mapupunta siya sa RER. Ano yung mangyayari sa RER? Dalawang processes yun sa RER. Hydroxylation and also glycosylation. That's why may nakalagay yan. Galactosyl, glucosyl. Galactosyl, glucosyl, glycosylation, addition of carbohydrate. And then from RER, mapupunta siya sa Golgi. No, Golgi apparatus wherein there is assembly of pro-collagen. Okay, and remember, tingnan yung structure ng pro-collagen, di ba meron siyang helical part, and sa lateral portion, merong non-helical terminal portion. Okay, and then, after that, ayan, may nakalagay, non-helical peptidase. Okay? Ayan. And then, what's next? Mare-release via via exocytosis. Okay? Mare-release itong pro-collagen. Okay? Now, wait up. After assembly, meron pang mangyayari. Tama. May exocytosis. Then from that exocytosis, available na siya sa extracellular space. Nandoon yung enzyme na peptidases. Tama? Nandoon yung enzyme na peptidases. Kaya yung enzyme na yun, it will cleave. No? It will cleave the registration peptides. Forming now the diba? Ang final product dyan is yung tropocollagen. Okay, kapag na-cleave na yung natira is the tropocollagen. And then, from that, ayan. Okay, so from that, after forming tropocollagen, it will now form cross-linking, ayan, forming a more strong, a more matured na fibril structure. So, ito na yun. Okay, may networking na. Okay, overlapping na yung mga collagens. Okay, yung mga tropo collagen to be specific. Okay, para mabuo yung mas matured collagen fibrils. Ayan, summary lang yan. You can just go over or go back to your step-by-step na notes. Okay, for the biosynthesis of collagen. So that concludes our discussion on the synthesis of collagen. Now, okay. So done with the synthesis of collagen. Okay, continue to have, we still have time. After learning the synthesis of collagen, it is also very important to know and to understand that there are possible na mga diseases, possible condition in the body wherein the underlying problem is the abnormal synthesis of collagen. Okay? And yung tawag doon sa mga problems or conditions wherein ang underlying cause is due to abnormal synthesis of collagen it is collectively known as collagenopathies. Collagenopathies. So, the following are the examples of diseases in relation to the abnormal synthesis of collagen. So, number one, we have osteogenesis imperfecta, or this is also known as the brittle bone syndrome. Okay? Brittle bone syndrome. Okay, wherein, yung nangyayari dito, may mutation. Okay? May alteration, may mutation na naganap involving replacement of the glycine, okay, with bulky side chains. Okay, with amino acids na merong bulky side chains. So, nare-replace yung glycine with amino acid na sobrang laki yung side chains nila. Okay? Question. Is presence of glycine very important for the formation of collagen? Yes or no? Yes, it is very important, diba? It provides flexibility. Bakit flexible yung characteristic ng glycine? Kasi this has a very small na side chain. Okay, this is the simplest actually na amino acid. So kapag may mutation sa glycine residue, that causes abnormality already sa synthesis ng collagen. So that causes osteogenesis imperfecta. Yan yung underlying problem. Okay, so in relation siya sa abnormality sa synthesis ng collagen. Next, we have Ehlers-Danlos syndrome. Okay, so for this condition, this is characterized by inheritable defects, in the collagen molecules. So, inheritable siya. So, it has something to do pa rin with the synthesis defects sa collagen formation, in which ang defective genes are the ones that encodes alpha collagen 1, pro-collagen N peptidase, or lysyl hydroxylase. Remember, these are all important na substances, enzymes. Very important sila na enzyme, okay, for the formation of mature collagen. Halimbawa, yung pro-collagen peptidase, diba? Na-mention kanina yung pro-collagen peptidase. Also, the lysyl hydroxylase. Ano yung importance nila? For pro-collagen peptidase, halimbawa, diba? This is an hydrolytic enzyme. Okay? Para saan? It cleaves, It is the one that cleaves the non-helical portion of pro-collagen. Diba? Naalala? It is the pro-collagen peptidases. Whereas, itong lysyl hydroxylase, It is also important for formation of hydroxyl-lysine, di ba? The HYL. Yan yung enzyme, di ba? The lysyl hydroxylase or lysine hydroxylase. So kapag may defect dyan sa genes na nag-e-encode sa mga enzymes na ito, that will cause Ehlers-Danlos syndrome. Okay, so just take note of the mutations na nangyayari or defects in relation to collagen biosynthesis. Next, we also have this condition, yung Menkes syndrome. Ayan, Menkes syndrome. This is also known as kinky hair, kinky hair syndrome. The problem with this condition, it has something to do with the lysyl oxidase naman. Okay? It has something to do with the enzyme lysyl oxidase. Kumbaga, yung nangyayari, inactive. Okay? Inactive yung enzyme na lysyl oxidase because inactive siya because of the deficiency of copper. Okay, remember, ang cofactor na kailangan ng lysyl oxidase, okay, is si copper 2+, diba? It needs the cofactor Cu2+. Therefore, kung wala or mababa masyado yung copper 2+, hindi ngayon ma-activate yung enzyme na lysyl oxidase. Okay, so absence, deficiency of copper, it affects the lysyl oxidase action. Kaya apektado overall yung cross-linking process. Okay, so yun yung underlying cause ng Menkes syndrome. And regarding Menkes syndrome, this usually affects males. Okay, it usually affects males, and again, it affects copper levels, mababa yung copper levels. It also affects the metabolism ng copper in the body. Okay, causing seizure, brain damage, weakened bones, weakened muscles, the organ shutdown, or failure to thrive. Okay, so yun yung problema ha sa Menkes syndrome. It has something to do with the inactivation of lysyl oxidase due to deficiency of copper. Somehow, you have encountered this at PChem 1, diba? Deficiency of copper, Menkes syndrome, okay, or the kinky hair syndrome. Next, scurvy. This is a condition which is caused by deficiency of the vitamin C. Okay, scurvy. Somehow you're familiar with this, scurvy, deficiency of vitamin C. Question, is vitamin C important in the synthesis of collagen? Is vitamin C important? Yes. For collagen formation, yes. Kapag depleted yung vitamin C, it will cause defective na hydroxyproline. Okay? Magiging defective yung mabubuong hydroxyproline. Okay? And even hydroxylysine formation. Okay? So kapag defective yung mabubuong HYP or HYL, that will make the collagen weak. Weaker ang mabubuong collagen, very fragile yung collagen ng mabubuong masisynthesize kapag defective vitamin C. Okay? Kumbaga kapag fragile masyado yung collagen, mabilis lang siya masira yung collagen na nabuo. Kaya ang tendency sa mga patient na suffer from scurvy, okay, mabilis magdugo yung gums. Kasi again, very weak, not stable yung collagen na nabubuo. Kaya madali lang mag-bleed yung gums nila. Okay, kasi repeat, weaker collagen yung nabubuo due to deficiency of vitamin C. Okay, may nakasulat dito, it is characterized, scurvy is characterized by bleeding gums, swelling of joints, poor wound healing. Ayan, kapag mababa ka sa vitamin C. Okay, and ultimately, kapag hindi yan kontrolado, sobrang mababa na, that will cause, ultimately death. Okay, so overall, done with collagen.
Now, let us proceed to elastin. This is specifically a connective tissue protein possessing a rubber-like properties. Okay, pag sinabing rubber-like, ibig sabihin, it can be expanded. It can be expanded. Okay, and when it is capable to be expanded, and then it can actually go back then to its original position, to its original form. Okay, that is why it is termed as elastin kasi elastic siya. Okay, elastic siya, yung characteristic niya. Okay, elastin is also found in different parts of the body. Just like in the lungs, very dominant in the lungs, also found in the walls of the blood vessels, and of course in elastic ligaments. Okay, so as stated, it is found in the lungs, it is found in the blood vessels. This is the reason bakit elastic yung blood vessels, and also lungs. Okay, they are able, diba, to expand. They are able to expand and then get back to their original composition or form kasi nga they contain elastin protein. So as mentioned, it can be stretched, no, stretched to several times, and they can be recoiled back to their original shape when they are relaxed. Now, regarding the primary structure of elastin, this is actually composed of primarily small and non-polar amino acid residues. I repeat, talking about the primary structure of elastin, it is composed primarily of small amino acids, at the same time those non-polar amino acid residues. So specifically, we have glycine, alanine, and valine. Diba? Recalling the side chains of this amino acids, glycine has hydrogen side chain, alanine has a methyl side chain, and what about valine? It has isopropyl side chain. Small side chains, non-polar at the same time. Okay, and because of that smaller sizes ng amino acids, that contributes to the flexibility of elastin. So yan yung isa sa mga reason bakit flexible yung elastin kasi nga in its primary level, primary structure, it is composed of smaller na mga amino acid residues. Okay. Also, this is rich in proline and lysine. It is also rich in proline and lysine. However, in the case of elastin, it contains little, very little derived amino acids like hydroxyproline and hydroxylysine. Okay, so little lang yung HYP, HYL niya, unlike collagen. Also, interchain cross links form desmosin residues. Okay, kapag nag-cross linking na yung mga amino acids on the present on elastin protein, that will form desmosin. Okay, so usually kapag sinabing cross-linking, okay, overlap na dyan yung mga mentioned amino acids on the primary structure of elastin. Okay. Now, with regards to desmosin, this is particularly an extensively interconnected rubbery network, okay, that can stretch and bend in any direction and will go back to its original shape, okay, when it is relaxed. Okay. Now, since it was mentioned earlier that yung elastin, okay, is present sa lungs, present sa blood vessels and other ligaments in the body. Discuss natin ngayon yung elastin in connection to that. Okay, particularly as you can see on this animation, you have there actually the elastin present in the lungs, okay, present in the lungs of a human. And of course, when we inhale air, diba? When we inhale air, hindi lang naman oxygen yung na-inhale ng isang tao. But there are also other toxic substances that we inhale. Okay? And kapag may toxic substances na na-inhale or may pollutants tayo na sumasama sa hangin that we inhale, the goal of the body is of course to remove, no, to remove out those pollutants, to remove out the toxic compounds. Okay, I repeat, as we breathe, okay, we are not only taking in oxygen but other pollutants as well. Okay, now good thing, kahit we take in pollutants, we have this what we call elastase. Okay, in the given figure, yung mga kulay puti dyan, imagine these are the enzymes elastase. Okay, elastase. Yung kulay puti. Then yung mga kulay yellow, okay, na bilog, okay, yan naman yung pollutants halimbawa. Okay. So, yung elastases dyan, elastase, yung enzyme, that is actually an enzyme that degrades or remove, okay, those pollutants, okay? Yan yung elastase. So, elastase, good thing, andyan siya sa lungs, okay, because it will remove off, okay, aalisin niya yung mga pollutants na nakapasok sa lungs. However, it must be realized as well that we have to regulate elastase. Okay? It has to be regulated. It has to be controlled. Okay? Dapat kontrolado yung action ng elastase. Kasi kapag hindi kontrolado ang action nito, after degrading these pollutants, after removing these pollutants, okay, it can actually destroy the lungs. Okay, particularly, it could destroy the elastin. Okay, it could destroy the elastin present in the lungs. Kaya nga 'di ba tawag sa kanya elastase, okay? An enzyme that could potentially okay, degrade elastin in the lungs. Elastin. So, meron siyang capability talaga na to target elastin protein. Okay, as an enzyme, elastase. Okay, so ang goal dito, para hindi niya masira, no? Para hindi niya masira yung elastin at the end, yung mga elastases enzyme must be regulated, must be controlled. And good thing we have certain substance, no, also present in the lungs that could help regulate this enzyme. Okay, and to regulate elastases, we have this what we call the alpha-1 antitrypsin. Okay, the alpha-1 antitrypsin. So, dyan papasok yung role ni alpha-1 antitrypsin. Okay, kasi it will be the one that will deactivate. It will deactivate elastases. Okay? After performing its useful job. Diba? Yung useful job that I have mentioned earlier ng elastase is dinedegrade niya ito, yung mga pollutants. Yun na nga, kapag naubos niya, na niya yung mga pollutants, okay, kapag hindi kontrolado yung action ng elastase, kapag wala na siyang makitang pollutants, it will then target elastin in the lungs. Okay? And that should not be maintained, no? Okay? Dapat pigilan po yan na action ng elastase, degrading elastin. Okay? So para mapigilan yung elastin degradation, alpha-1 antitrypsin, okay, papasok siya, okay, sa action, sa eksena. Okay, so based on what I have discussed na concept, we can really say that alpha-1 antitrypsin sa lungs is really very essential. Okay, it is very essential. Okay, kasi nga, nakalagay dito, di ba, without antitrypsin enzyme, okay, kapag wala yung antitrypsin, okay, the elastase will continue, will continue its action. It will consume anything in its path, including the healthy lung tissue. And ano yung meron sa healthy lung tissue? Meron yung elastin. Okay, tatargetin niya yung elastin. Kapag walang antitrypsin, walang magkocontrol sa kanya, walang magde-deactivate kay elastases. Okay? However, ang problema, may mga tendencies, may mga cases, okay, or may mga ginagawa yung tao or habitual act yung mga tao na nagkukos ng pagbaba ng antitrypsin. Okay? I repeat, may mga cases, may mga tendencies wherein pwedeng bumaba ang antitrypsin in the body. And one of the major reason why antitrypsin may be depleted in the lungs, it's because of smoking. Okay, it's because of smoking. This is one of the major reason why antitrypsin may be depleted. Okay, it may be depleted because of smoking. I repeat, this is one of the major reason, okay, na napapababa sa lungs yung antitrypsin. Okay, therefore, based on the concept discussed earlier, kapag mababa yung antitrypsin or without antitrypsin, syempre, hindi na ngayon kontrolado or regulated yung action ng enzyme elastases. Okay? Kapag mababa yung antitrypsin, elastase could destroy a healthy lung tissue. Okay? And particularly, it will target protein elastin. Okay? Kaya, take note, pag may chronic smoking, pwede magkasakit yung tao. Pwedeng magkasakit ang tao sa lungs. And ang tawag dyan, okay, ang tawag po dyan is emphysema. Okay? Ang tawag sa condition na yan kapag may deficiency of alpha-1 antitrypsin sa lungs leading to destruction of alveolar walls in the lungs, that is called emphysema. Okay, emphysema. So, emphysema, you can just read over this definition. So, yun lang yung concept niya. There is deficiency of alpha-1 antitrypsin. Okay? So, kung ang problema sa emphysema, mababa ang antitrypsin, so what would be the recommended treatment? Mababa ang antitrypsin, therefore, we simply administer alpha-1 antitrypsin in the body. So, pwedeng mag-administer ng synthetic or exogenous antitrypsin. Okay? Isosupply niya lang yung deficiency, okay, na meron sa tao. Mababa yung alpha-1 antitrypsin. And of course, kapag kapag may alpha-1 antitrypsin na sa katawan, mayroon nang de-deactivate sa elastasis. Okay, so hindi na masisira ngayon yung elastin. Okay, so para maiwasan yung emphysema or emphysema, you can have administration, okay, or to treat emphysema, you can have administration of alpha-1 antitrypsin, an exogenous one or synthetic, or the best prevention is really to prevent smoking, iwasan na lang yung smoking habit. Okay. Now in this figure, this is another figure that discusses the pathogenesis of emphysema. So halimbawa, you have here tobacco. Okay, so halimbawa yung tao has a smoking habit, so you have that figure or picture of a tobacco. And again, with smoking, ano yung napapababa na substance? Bababa yung alpha-1 antitrypsin. Napapababa niya yung alpha-1 antitrypsin. Kung wala or mababa yung antitrypsin sa lungs, that will further activate the action of elastasis. Okay? Hindi mapipigilan or hindi kontrolado ngayon yung elastasis. Kaya, it will target the elastin in the lungs, causing tissue damage in the lungs. And what do you call that condition? That's emphysema. That's emphysema. Okay, that's the end for elastin. Okay, so far we have discussed already two. Okay, fibrous protein, we have collagen and elastin. Okay, the last example of fibrous protein is keratin. Okay, keratin or alpha keratin, this is a protein that forms tough fibers. And these are commonly found in the hairs and nails and even in the outer epidermal layers of the mammals. Okay, so this is a common knowledge. Yung present the protein in our nails, in our hair, it's keratin. Okay, now talking about the primary structure of keratin, this is actually composed of cysteine residues. It is composed of cysteine residues. So kapag cysteine residues, okay, segments of cysteine residues that will probably form disulfide bonds. Okay, may presence of disulfide bonds. Yan yung most common features sa primary structure ng keratin protein. Okay, this is also the reason why hairs, okay, and the nails are considered water insoluble. Okay, water insoluble sila. Okay, kasi as you can see on the primary structure, it is composed of cysteine residues, and forming disulfide bridges, it only suggests that these residues are non-polar. Okay, non-polar residues. And we all know that water is polar. And if cysteine are non-polar, therefore, water cannot dissolve keratin, therefore, or nails. Okay, kaya nga 'di ba kapag naligo tayo, hindi naman natutunaw yung hair natin, hindi natutunaw yung nails kapag naligo tayo using water. Okay? Kasi nga what our hair and our nails made of, it is made up of non-polar residues, cysteine residues. Okay? Now with regards to hair, talking about hair, 'di ba? We have hairs nga kinky, meron tayong straight hair. We have straight hair, we also have kinky hair. Okay. The reason why may mga kulot, okay, na form of hair, it is because sobrang rich ng disulfide bonds na makikita sa buhok. ng mismong tao. Okay? So, yung hair niya, okay, yung keratin niya, it is made up or rich in disulfide bonds. Okay? With sustained residues or formation of disulfide bonds that causes kinks, okay, or formation of kinks sa buhok. Okay? And might be when you do hair rebonding, pwede yung sabihin sa parlorista, okay, na you can have reduction of disulfide bond, no? When you do rebond, that is synonymous lang po sa process na reduction of disulfide bonds. Okay, reduction of disulfide bonds. Kasi nga kapag kung dahil sa disulfide bonds, kulubot yung buhok, we can simply break, no? You simply break those disulfide bonds that will lead to straightening of the hair. So that's a reduction of disulfide bonds. I don't know what is the logic bakit it is termed as rebonding kasi pag rebond, 'di ba? Binuo yung bond, but what we are actually doing kapag rebonding, we are breaking the bonds, no? Breaking the disulfide bonds. Okay, so I repeat, yung keratin, alpha keratin, this is most commonly found in the hair, in the nails, and these are considered water insoluble. Water insoluble because of the sustained residues that are said to be non-polar.